Great. Thank you for that very generous introduction, Alex. I'm really excited to be here today. I'm just rearranging a few windows. There we go. So before we begin, I would like everyone to close their eyes because we want to activate our sense of sound. What we just heard was the sound of a rainforest in Eastern Africa. And that sense of calm that we experience when we're in nature, that's no coincidence, right? That's by evolutionary design. We evolved in an ecosystem very similar to this one, where those sounds of insects, birds, mammals, those indicated the possibility of food, medicine, all those kinds of resources that we needed to survive. That biodiversity is still key to our well-being, the future. Our survival as a species depends on that biodiversity. I've been fascinated by biodiversity, and I've dedicated my entire career to understanding the intersection between biodiversity and climate change. Even though biodiversity and climate change, we know they're interconnected, they've historically been studied by very separate disciplines. As an example of this, two of the largest intergovernmental bodies, the IPCC, the Intergovernmental Panel on Climate Change, and the IPBES, the Intergovernmental Platform on Biodiversity and Ecosystem Services, these two bodies have operated in isolation until just last year. And I think I'm naturally drawn to intersections between disciplines, between ideas, because I've spent my entire life in an intersection. I grew up in an intersection of culture, language, gang lines. I grew up in what's known as the South Texas Chicano tradition, where everyone was all or part Mexican, and moving between English and Spanish played a prominent role in everyday life. I tell people I was not a minority growing up. I became a minority when I left home to go to college. And even as in college, as an undergrad, I continued to exist in an intersection. As a federal work-study student, I spent the academic year collecting Lepidoptera tissue for DNA barcoding. This is at Guelph, this image on the left, with Dan Jansen. And then I would spend the summers in Costa Rica cataloging caterpillars, their host plants, and parasitoids, and learning alongside parataxonomists in Spanish. But what I was learning about science from English language textbooks often contradicted what I would see in the field working in Spanish alongside parataxonomists. And this movement between learning about science in English and doing science in Spanish led to a really important revelation in my career. I realized that I had this ability to identify these overlooked ideas and assumptions in ecology. And that was because I spent an extraordinary amount of time in the field, and I'm an outsider. As an outsider, I don't feel obligated to subscribe to traditional ideas or ways of doing science. And And this ability to see and do things differently has become a signature of research in my lab. This is also a great example of how diverse perspectives, no surprise, lead to innovation and new insight. Today, I'll provide 3 examples of how we challenge dominant paradigms and assumptions at the intersection of biodiversity and climate change. I'll start by describing the role of soils as an overlooked driver of biodiversity. Next, I'll demonstrate how we use data science to fill in the supposed geographic gaps in biodiversity patterns. And I'll end by describing some new initiatives that we're working on to understand biodiversity responses to climate change from local to continental and global scales. So one of the most dominant views in ecology has been that climate controls the geographic distribution of biodiversity. This is an idea that is 2,000 years old. It dates back to the 3rd century BC, back to Aristotle and Theophrastus, and in large part still dominates modern macroecology, where biodiversity patterns on Earth are primarily attributed to climate at these macro scales. But there's this growing recognition that geodiversity is an important control on biodiversity in some disciplines. So geodiversity is defined as landforms, topography, soils, bedrock, water features, any kind of enduring landscape feature. And the role of geodiversity as a control of biodiversity is so prominent in some fields that it's become a key component of conservation strategies. This is a strategy called conserving nature stage. The role of geodiversity as a predictor of biodiversity can also be seen across the world's biodiversity hotspots. Almost 70% of biodiversity hotspots across the world occur on unusual soils. So these are things like your limestone, granite, serpentine, gypsum, and this effect is especially prominent on serpentine soils. So serpentine soils have fairly unique characteristics. They tend to have high concentrations of heavy metals, which is usually difficult for plant growth. They also tend to be nutrient poor and have low water holding capacity. And these unique features create obvious vegetation changes in the landscape, what's classically known as the serpentine syndrome. So at this image that you're looking at here, the serpentine soils in the foreground, they're dominated by grasses and herbaceous plants. And the non-serpentine soils in the background are dominated by trees, or vegetated by trees. And while the role of geodiversity is recognized in disciplines like conservation science, it's still very much neglected in disciplines like macroecology. So with this in mind, I led a synthesis paper in which we outlined 4 key processes by which soils drive biodiversity at these global scales. We focused on speciation, dispersal, ecological drift, and niche selection. And the idea that soils are important isn't new, but what we've done is describe ways in which soils are important in a way that really hadn't been synthesized in the literature up to that point. As part of this process, we ended up mapping the global distribution of serpentine soils on this map, both in gray and in black. And it became clear that most of what we know about unusual soil systems comes from temperate latitudes. And we realized that a lot of the hypotheses developed on unusual soils in temperate latitudes just weren't relevant in the tropics. And in fact, tropical serpentine ecology contradicts our understanding of unusual soils. So let me give you an example of what I mean by this. This is a classic temperate serpentine grassland in Northern California. This is a system dominated by grasses very xeromorphic vegetation. This is classic serpentine syndrome in the sense that the plants are very short statured and they have small, thick leaves. But in the wet tropics, it is not uncommon to find gallery forests that have formed on top of serpentine substrates. This is an image from Puerto Rico. It's very clear the forest structure is quite different. The processes are quite different. Those soil-climate interactions are even more obvious in places like Costa Rica, where this entire serpentine massif, this is La Península de Santa Elena, it's entirely blanketed in clouds for most of the year. Below cloud line is that classic serpentine syndrome, what we call chaparral, and above cloud line, textbook moss-covered tropical cloud forest. This work led to a second synthesis group to better understand the plant ecology of tropical serpentine ecosystems, because we know that theories like the classic serpentine syndrome were developed in temperate systems with fairly moderate temperature and low to moderate precipitation. And these ideas aren't relevant to tropical ultramafic or serpentine ecosystems because these systems experience much warmer temperatures, they experience a wider range of precipitation, and this combination of warm temperature and high precipitation can act to ameliorate the serpentine syndrome that's reported in temperate latitudes. This led to a second synthesis paper. And this was such a wonderful collaboration because we were very intentional about inviting people, early career researchers living and working in these serpentine systems around the world. And it's inspired a few other papers that we're working on now. In this paper, we showed that tropical serpentine systems are diverse, and that diversity is driven by interactions between soils and climate. So something that we're working on right now is collating a global GIS database for all serpentine outcrops around the world. And at first glance, this doesn't seem very exciting, but what's exciting about a database like this is that it enables the types of macro scale questions combining satellite imagery and fieldwork to understand those soil climate interactions on biodiversity at a global scale, at a scale that really hasn't been possible to date. This type of database also generates new ideas about the conservation of biodiversity hotspots in a global context, given that most biodiversity hotspots occur in areas of these unusual soils like serpentine. So in this first example, we explored the overlooked role of soils as a driver of biodiversity, which has led to some really nice collaborations. And in some ways, maybe challenging who gets to participate in these large macro scale, macro ecological working groups. In this second example, I'll describe how we identify assumptions through data science. For this project, we really wanted to focus on a topic that was unresolved and that inspired a lot of discussion and debate in the literature. And this is definitely true of the insect apocalypse. And global population decline of insects is very concerning. It threatens the global food supply, as well as cascading consequences on food webs and trophic interactions. But despite the publications and media coverage on this topic surrounding insect declines, there's still a lot we don't know. For example, most reports of insect declines have been conducted at small scales or are focused on a single species or groups, making generalizations elusive. And most studies of insect decline have been temperate, even though most insect diversity is tropical. And a lot of high-profile scientists claim that the reason why we don't yet understand the full picture of global insect decline is because data on tropical organisms is lacking. And in my lab, we challenge that idea. It's simply not true. Data on tropical organisms may be lacking in English language publications or in English language databases. But there's a huge treasure trove of data from the tropics in places like the gray literature, including dissertation theses, regional publications, and especially natural history museums. Mining this data allows us to use modeling approaches to fill in those supposed geographic data gaps, especially in the tropics, and they can also generate testable hypotheses of how entire ecological systems respond to climate change. Butterflies are a great focal group for modeling approaches and filling in those data gaps in the tropics because butterflies as ectotherms, in theory, they should be responsive to things like temperature change. They're also charismatic and so receive lots of public attention. You'll often see very professional hobbyists. And they're also well studied. So we know quite a lot about their natural history, which is exactly what you want for a group in which you're doing a purely modeling approach. You want to be able to compare the modeling results to known biology. In many tropical regions, like the Caribbean biodiversity hotspot, there's a long historical record of butterfly collecting in this region, as well as reliable identification guides. The Lepidoptera fauna is especially well documented in places like Puerto Rico. Beginning in the 1940s, what's now the American Museum of Natural History was pretty much a very colonial 40-year-long inventory of the flora and fauna of the island, including leps. And because of this colonial legacy, there's always been a strong focus on taxonomy and not so much synthesizing those biodiversity patterns across the island. So we wanted to use this approach to address, to fill in those knowledge gaps and address two questions. First, we were interested in understanding just where are the diversity hotspots across the island. This first question is an important question to guide conservation priorities. Conservation group that we work with on the island, Para la Naturaleza, they're very interested in this type of data. And the second type of question we wanted to address is whether climate and functional wing traits mediate species distributions. And this is an important starting point for understanding potential climate change impacts. The environmental variation across the island, here you can see elevation. It creates this natural experiment for understanding diversity and climate change on the island. In terms of diversity, to me, it really wasn't clear where those diversity hotspots should be. Based on 20 years of working in Costa Rica, I thought, well, in mainland tropics, the wetter forests tend to be more species rich, and those tend to be at higher elevations. So I thought, okay, maybe that's true here as well. In terms of wing traits, there are two hypotheses that can guide our understanding of trait diversity patterns across the island. The first is Bergman's rule, which predicts that a body size should be larger in cooler habitats. And the second is the thermal melanism hypothesis, which predicts that organisms should be darker in cooler habitats, both to aid in thermoregulation. So the idea is that darker colors absorb more thermal energy. The same concept applies to body size as well. If you have larger surface area, you can absorb more thermal energy. So based on these two predictions, we expected a larger, darker species to occur at higher elevations, and those smaller, lighter species to occur in the lowlands where it was hotter and drier. To test these ideas, we first had to digitize all the butterfly collections on the island. So we led a island-wide digitization campaign with the help of many of the museums, as well as iDigBio, which is an NSF-funded national consortium of museums that are digitizing their collections. We also had the help of incredible students, and we digitized something like 6,000 specimens for almost all the butterfly species known on the island for a fraction of the budget of most iDigBio projects. With those digital images, we then extracted wing traits using image analysis software. And I had an undergraduate student complete a preliminary test of this methodology on the non-migratory Puerto Rican monarch before we attempted to work with all of the butterfly species. We focused on wing length of the forewing depicted in the image, as well as wing color in that 1 centimeter region close to the thorax. This region has been shown to be important for thermoregulation. And then to address this question of where are the diversity hotspots on the island, we first calculated species distribution models for all the butterfly species. Now, this is a really complex process, so I won't go into the fine details here. We also recognize that a lot of the methodology is evolving in the past 5 years, so we used some of the more conservative thresholds and criteria. This process begins with collating records, including the ones we digitized, but also from sources like GBIF. Then you want to process those records and reduce spatial bias. You want to collate your environmental parameters from things like world climb. Then the actual modeling, we use 3 types of models: Maxent, which is the kind of old school approach, random forest, and BioClimb. So this is an ensemble approach. And then we took all the individual species distribution maps and stacked them to generate an island-wide diversity map. And then we did some post-processing analyses to understand how diversity is mediated by climate and wing traits. So to answer this first question, just where are the diversity hotspots on the island? Remember, my naive answer was, well, in those higher elevation rainforests, but we found something opposite. Diversity or species richness was highest in lowland tropical dry forests. And this turns out to be a similar pattern that's been found for bird diversity across the island. And for birds, the working hypothesis is that, well, there are fewer of these coquilles or amphibian insect predators, which leads to a greater population size of insects, which in turn supports more birds. So this may be true for butterflies as well. There may be fewer butterfly predators in dry forests. To answer this second question, how does climate and traits mediate species distributions? Remember, we expected wings to be larger and darker at those cooler, higher elevation habitats, and smaller and lighter in the lowlands. We found something slightly different. Wing size seemed to follow Bergman's rule, but wing color was opposite to predictions based on the thermomelanism hypothesis. Instead, we found species were darker at those lower, warmer habitats. And so we wanted to pair this purely modeling approach to sampling that we had done in the field immediately following Hurricane Maria. And if you ask anyone about their memory of this experience, myself included, one of the things we will probably remark on is how an island that was 100 different shades of green turned to a single shade of brown in a matter of 12 hours. And this complete island-wide defoliation event created fairly homogeneous conditions in the understory, right? Very high light, open canopy, very hot conditions. And we followed butterfly recovery immediately following the hurricane. Based on these same 2 hypotheses, Bergman's rule and thermal melanism, we kind of expected to find the same thing, that smaller, lighter species should dominate those hot, open habitats immediately following the hurricane. But then as forest recovery occurred, we expected to find an increased abundance of those larger, darker species. However, we observed this same pattern as we saw in the species distribution modeling. Shortly after the hurricane, the smaller, darker species dominated, followed by an increase of abundance in those larger, lighter species. So together, these two studies, the species distribution modeling and the butterfly recovery post-hurricane, indicate possible energetic trade-offs between growth, thermoregulation, and defense. We know that melanin is important for thermoregulation. We also know it's important for things like immunity. And we also know that it is a pretty energetically expensive pigment to manufacture, which could result in these types of trade-offs between wing color and wing size. An alternative explanation is that maybe what we're seeing is an effect of seasonality, where those smaller, darker species dominate in the winter, in the cooler winter, whereas larger, lighter species dominate in the summer. Or prey visibility, that those larger, lighter species were more visible to prey immediately after the hurricane against that very brown backdrop, and got picked off faster or easier, whereas those smaller, darker species were better camouflaged in the browner environment. What excites me about collections-based research is this great potential for collaboration across disciplines, across countries, and it also creates a lot of opportunities for students. Collection-based research has been very productive in my lab, even though we haven't had very large pockets of funding earmarked for it. Just this past summer, we had 3 collections-based research internships in my lab, ranging from a DNA barcoding project to outlining a century of LEP research in Puerto Rico, making that gray literature accessible to an English language audience. And I also had a student visiting different herbaria to measure plant trait variation across Caribbean dry forests. So in this second example, I demonstrated how we can leverage collection records in the tropics to fill in those geographic gaps in our understanding of biodiversity and potential responses to climate change. In lots of ways, our findings also challenge these dominant predictions related to thermal melanism. And we also establish research priorities, focus on LEP ecology, and moving forward on the island. And in this final example, I'll describe some of the newer stuff that we're working on that continues to leverage this creativity and diverse perspectives to measure the consequences of changing biodiversity from local to global scales. A lot of the more recent work in my lab focuses on mountains because mountains are biodiversity hotspots. They're centers of endemism and speciation. They're also key for human health. Almost 60% of all humans on Earth get their water from a mountain source. And they're also important climate change indicators and natural laboratories for understanding climate change. And so I've recently launched an initiative called ALTA based on funding from my career award. ALTA stands for Altitudinal Transects Across the Americas. And the goal of ALTA is to demonstrate that it's possible to design inexpensive cross-site biodiversity monitoring. And this is especially important for regions that are undersampled and are experiencing accelerated rates of climate change. So on the map, I'm showcasing a few of the places where I've either sampled in the past or we're sampling right now in Virginia and the Great Smokies. And I'm also showing a few mountains that I have my eye on as potential wish lists, and I'm figuring out how to approach potential collaborators in these regions. Across all of these ALTA sites, we're measuring things like forest dynamics. Next year, we'll be launching a seedling census because the understory is this neglected environment. We don't know a whole lot about what's going on in the seedlings, even though they indicate they're the first indicator of climate change. I'm not a plant physiologist, but I would love to work with one because a lot of literature right now is emphasizing that both thermal tolerance and drought tolerance of plants is likely linked. And finally, we're also starting to use remote sensing data to understand vegetation changes at regional to continental scales, and eventually to connect that plant physiology to the macroecological changes. Each ALTA site has a similar design of these forest dynamics plots situated across elevation, but each research site has its own research or science flavor that's in part guided by the land managers of each region. So in this last example, I'll focus on one of our ALTA sites in northwestern Costa Rica, where we're especially interested in identifying the ecotone between tropical dry forests depicted here in brown and tropical wetter forests in green. And this is because we're focusing on the ecotone because this is where a lot of the changes in response to climate change will happen first, because ecotones are windows into both organismal to biome scale climate change responses. Ecotones have a special place in the foundation of ecology as a discipline. The term ecotone actually preceded the ecosystem concept. And what's really interesting about ecotones is that they're both spatially diverse and temporally dynamic. They control the flow of energy and organisms across landscapes. They're typically determined by species' physiological limits, which makes them sensitive global change indicators. And there's 3 types of ecotones we know quite a lot about at a global scale. The first is this shrub encroachment into grasslands, which is this global phenomenon. The second is encroachment of salt marsh into uplands, creating these ghost forests. And for the aquatic ecologists in the audience, the deep chlorophyll layer is an important ecotone that's impacted by warming waters and shifts in nutrient availability. And I think we should be studying ecotones in many other contexts across scales, but methodological challenges have been difficult to overcome, which has limited our ability to use ecotones as indicators of global change. And this is because ecotones are inherently variable, which in the past meant lots of ground-based measurements, creating these logistical constraints for detecting and monitoring ecotones. And because of these, consensus on definitions and methods have been elusive. But modern remote sensing and quantitative techniques, they offer solutions for detecting and monitoring ecotones through space and time, as well as for understanding those landscape responses to climate change. And we also hope to apply this approach to monitor shifts in entire biomes. Together, tropical dry and wet forests make up the majority of tropical forest area, but we don't know a whole lot about the ecotone between these 2 biomes. As a tropical biologist, you usually tend to work in one system or the other, not necessarily across both of them. So in my lab, we specialize on finding those underexplored interactions between ideas, between disciplines, as related to biodiversity and climate change. These three examples demonstrate how this creative, and I've had it called unorthodox and subversive approach, I don't know about that, can lead to insight into some of the biggest challenges, not just facing our discipline, but the world. And in this first example, we saw how soils are often neglected in discussions on global biodiversity patterns. And we use serpentine, we positioned serpentine as a new macroecological model to link climate, soil, and biodiversity. In the second example, we use data science to fill in those supposed geographic shortfalls in the tropics. We used a modeling approach to outline a research agenda for LEP ecology on the island. And in the last example, I described our new initiative called ALTA, and we looked at how remote sensing could help us detect and monitor ecotones in ways that would have been intractable from fieldwork alone. I'm always looking for collaborators. We're just launching these new ALTA initiative. So if these approaches excite you, if you identify as a creative, then please email me. I'm happy to continue this conversation later. And I think we have plenty of time for questions now as well. So thank you.
That was awesome.